Right. Good afternoon, everybody. Thank you so much for joining us today. My name is Stephanie. I will be monitoring things here on the back end. So if you have questions, you can put them in the Q&A or any comments. You're more than welcome to use the Q&A or the chat. Both should be activated. It's my pleasure to introduce our second talk in our neuroimaging summer series, where we are showcasing the cutting edge research of the neuroimaging faculty and associates at the Michigan Alzheimer's Disease Research Center. Today, I am pleased to introduce Roya Homayouni, who is a postdoctoral research fellow at the Institute of Gerontology at Wayne State University. Roya completed her PhD in behavioral and cognitive neuroscience at Wayne State University in the winter of 2024. And immediately after, she started her postdoctoral training at Healthy Brain Aging Lab under the supervision of Dr. Anna Doherty. Roya's research is focused on delineating the lifespan age trajectories of hippocampal subfields. Volumes of their memory correlates while exploring the nuanced sources of individual differences in age trajectories. Today, she's going to talk about her research, particularly the use of high-resolution MRI methods for hippocampal subfield volume tree to reliably answer her research questions. And with that, I will turn things over to you, Roya.
Thank you very much for the introduction. It's a privilege for me to be here and talk about my research and share some of the findings. Can you see my slides now?
Yes.
So in today's talk, I will talk about age-related studies of hippocampal subfield in vivo and the importance of this research line in the field. I will then talk about implementing high-resolution neuroimaging technique for hippocampal subfield volumetry. For the purpose of this talk, I will go over more details on the method section, just so highlight the most important methodological consideration for a reliable practice in hippocampal subfield investigations. Lastly, I will share with you some of our findings on hippocampal subfield based on volumetric measures, noting that all the findings that I will discuss today is based on healthy population, but anywhere possible in my talk, I will highlight the importance and implications of these findings for better understanding of clinical populations. So I will start with episodic memory. That is cognitive domain that I am most interested in. Episodic memory is an integral aspect of cognition that enables us to remember daily experiences. Episodic memory is characterized with high level of enrichment and complexity, for example, where, when, what happened, and also with high specificity and distinctness, so that you could remember similar memory events with low interference. As an example, with episodic memory, you could not only remember that you have parked your car in a certain time and location, but also separate that from similar memory events and in the past and in the future. With that, episodic memory is fundamental for our daily and functioning and scholarly life. We accumulate knowledge and make informed decisions. The ability of episodic memory is not constant across ages, and it is substantially changing across the lifespan. In this schematic figure, you see the stages of maturation, where episodic memory becomes more detailed and specific across child development, remains relatively stable from young adulthood to middle adulthood, and later in life, we see memory is characterized with high interference and low discriminability. Just noting that this level of memory decline is not at a clinical level. This trend that you see here, and I discussed, it is suggesting that age is a significant predictor of memory ability across individuals. However, it is not the only one, because when we look at the individuals of same ages, we still see that memory ability is significantly different. And in some cases, it actually can be at clinical level, such as the cases we see in patients suffering from Alzheimer's disease, where their memory ability is highly impaired, and that negatively impacts their daily life. So 2 questions can be asked here. First is, what explains the memory ability across different ages? Or what are the nodal correlates of episodic memory development across the lifespan? Another question is, why individuals are different? What factors contributes or impacts memory ability above and beyond effect of age? To answer these questions, attention has been drawn to the hippocampal structure that is fundamental neural substrate of episodic memory. You see hippocampus in sagittal view that is located in the temporal horn of lateral ventricle. The same hippocampus that is dissected out, and you have a bird view of that structure. And the same, again, hippocampus that is cut coronally. In this coronal view of the hippocampus and with your naked eye, you still see some layers within this structure. And in fact, hippocampus is not a unitary structure, and it is composed of different subfields, dendrite gyrus, CA regions with 3 sectors, and cerebellar area. In this figure, you also see interhyoid cortex that is not part of hippocampus, but it is usually studied together with subfields as a main gateway to input hippocampal circuitry. Subfields are highly interconnected, but they are also distinct in their structure and function. You see a comparison between pyramidal neurons of CA regions and granule cells of the dendrite gyrus. You see that these neurons are different in their density, size, dendritic arborizations, and in their layout. Subfields are also different in their connectivity pattern, as illustrated here with different colors of arrows in this schematic figure. And also they are different in activity level, as demonstrated here by the number of cubes. You see high level of CA1 activation as compared to dendrite gyrus. Another difference is distinct blood pattern of blood supplies. For example, CA1 is the region that is suggested to be less vascularized as compared to other subfields. So growing evidence coming from computational modeling is suggesting that subfields likely have differential function specializations in supporting episodic memory. For example, dendrite gyrus is supported to support memory specificity, and CA3 is proposed to support memory complexity and enrichment. As I discussed earlier, these are 2 major characteristics of episodic memory that change substantially across the lifespan. That is suggesting that probably subfield investigations are key for better understanding of neural correlates of episodic memory across lifespan. Subfields also are different in their sensitivity to lifestyle, environmental factors, sleep, and also they show differential vulnerability to a wide range of neurological and psychiatric diseases. For example, CA1 and cerebellum are shown to have early vulnerability to Alzheimer's disease. This is again suggesting that for the researchers who are investigating the sources of individual differences, they may find their answer if they can study those factors in relation to subfields. So inspired by the findings coming from computational modeling, animal and human postmortem studies, there has been ongoing effort to investigate hippocampal subfield in vivo, and advances in high-resolution neuroimaging technique has made this possible. You see here the number of publications in PubMed over the last 2 decades that use one of MRI technique to characterize hippocampal subfield structure and function in human population. One of the most popular and commonly used MRI technique to characterize hippocampal subfield structure is brain volumetry. That is obtained either by T1 weighted image that you see on the left or T2 weighted image that you see on the right. you see that hippocampal structure is nicely visualized in these high-resolution images. So in my study, I use high-resolution MRI to study hippocampal subfield volume in healthy population. I aim to characterize a trajectory of hippocampal subfield volumes across entire lifespan. My overarching goal is to understand the subfield contribution to memory development, to ultimately better understand neurocorrelates of episodic memory development across the lifespan. Age-related studies of hippocampal subfield volumes across the lifespan would also allow me to assess and identify the factors that impact subfield volumes at different stages of life, about and beyond effective age. That would ultimately open up the possibility for potential interventions for the benefit of healthy aging and development. And as I mentioned earlier, subfields are also involved in a wide range of neurological diseases. If we could establish healthy age trajectory for subfield volumes across lifespan, now we could use it as a benchmark, and that we could use it to compare volumetric differences against that baseline to identify potential deviations for the benefit of early diagnosis. So in this section, I'm going to talk a little bit the methods that we are using. For hippocampal subfield volumetry, we use T2-weighted proton density turbo spin echo that you see an example here for a 5-year-old male participants. This image is a partial brain coverage, which means we only focus on medial temporal lobe to fully cover the longitudinal axis of hippocampus. This partial coverage explains why this part of brain is not visible. T2-weighted image specifically offers the benefit of better visualizations of anatomical landmarks that are critical for hippocampal subfield segmentations. For example, here you see comparison between T2-weighted image and T1-weighted image. The landmarks that we use for subfield segmentation, these narrow dark band within and outside the hippocampus are pointed with this arrows. And you see they are nicely visualized in T2 as compared to T1. In T2 also, you see you have better morphometry and better visualizations of the width of the subfields, as you could see here in bottom row. In our lab, we also collect high-resolution T1-weighted MP range that is used for volume estimation of a cortical region and also intracranial volume. So T2-weighted images are collected on coronal plane and perpendicular to the longitudinal axis of hippocampus. Here I have added, I use a T1-weighted image on sagittal view just to explain image acquisition. And I added this line to represent longitudinal axis of hippocampus. So you see that image acquisition are oblique to this, would be oblique and perpendicular to this line. We collect slices for in total of 30 slices with 2 mm slice thicknesses, which means that if you are moving back and forth one slide on the T1-weighted image, T2-weighted image, you now are jumping 2 mm on the longitudinal axis of hippocampus. Here is the example of T2-weighted image on coronal view that is representing this oblique acquisition oriented perpendicular to the longitudinal axis. Our in-plane resolution is 0.4 by 0.4. That is considered high resolution, and it is typical. This describes the dimensions of every single pixel in this coronal view. I have added this scale bar that is representing 1 centimeter, just so show you the small sizes of subfields and also landmarks. And to just emphasize the necessity of subvolumetric investigation of hippocampal subfields. So after image acquisition, we segment hippocampal subfields using a protocol that is based on geometric heuristic. This protocol gives us volume estimates for 3 ROIs, as you see here, on the body of the hippocampus with approximate ranges specified here. The body is defined based on anatomical landmarks that are different across individuals, but usually it ranges from 7 to 9 continuous slices. This protocol also gives us volume estimate for interhyoid cortex that is estimated 6 continuous slices anterior to the first slice of body. We require subfields when manual tracing. We require each brain to be traced by 2 reliable raters that are randomly assigned for brain slices, and they are blind to the demographic information about that participant. This particularly guards against any potential bias that raters may impose on delineating subfield volumes. So over the last couple of slides, I discuss our practice for reliable investigations of hippocampal subfields. But the question here is the method really and statistically reliable? This is particularly important in the context of age-related studies that is based on longitudinal investigations and also multiple time assessment. When you have high reliability, now you could attribute volumetric changes between time points to developmental changes rather than measurement error. After all, there are a lot of factors that can be different in repeated measures MRI, such as repositioning the person in the scanner or behavior differences across sessions, or even habit of manual tracing across raters. In addition to high reliability, we also want the method to be sensitive to be able to detect nuanced individual differences. As repeatedly discussed in the field, and we also show it in our meta-analysis published last year, that effect sizes attributed on the age effect on brain measures are very small, and therefore we need a highly sensitive method to be able to detect that. We asked these questions a few years ago, and the results was published in this paper. Just to summarize those findings, what we did in that study, we collected two-weighted image for 28 participants from ages 7 to 20. And after a month, we replicated image acquisition, this on the same participants. The reason behind this 1 month delay was that we didn't expect significant developmental changes over this delay. Therefore, volumetric differences between 2 visits would have reflected measurement error or factors other than developmental changes. We manually segmented subfields based on our longitudinal protocol and assessed interclass correlation for left, right, and total volume of 4 ROIs with the cutoff 0.85. Our method showed an excellent consistency or reliability between 2 visits, with ICC measures being higher than 0.89, suggesting that measurement error is less than 11 or 10%, which is considered an excellent level. Importantly, our estimates were not biased based on size of subfields, age of the participant, sex, or hemispheres. To assess the sensitivity of the method, we replicated the same research design with different sample, now with 2 years delay. Here we expected actually developmental changes. And with wide age range that you see here, we also expected individual differences in the pattern of the change. If the method is sensitive, then it should identify significant variability in the change pattern. And actually, when we conducted bootstrap variance analysis, we found significant variance, as you see here, indicated by non-overlapping 95% confidence interval that were not overlapping with 0. In addition to direct implications of high reliable measurement in answering research question, in that paper, we also discussed the practical application of high reliable measurement in research design. We particularly discussed that measurement reliability optimizes statistical power and precision in hypothesis testing. For example, you see here number of participants that are required to achieve the power of 0.85 to be able to detect estimates of mean change to statistical significance, depending on the effect size and measurement reliability. For example, if you want to detect the effect size of 0.3 that is considered small, and your measurement reliability is 0.70, now you need 225 participants. That number drops to 125 participants if you have high reliable measurement of 0.90. This is important, particularly in the context of age-related studies that have small effect sizes. We also have sample attrition in longitudinal design, and also high cost of MRI technique. In that paper, we also discussed that if you have high measurement reliability that you see in blue color, you would have smaller standard error of the measurement and therefore narrow confidence interval. That's suggesting that you have more precise estimate of parameter. For example, if you have age trajectory for healthy population with narrow 95% confidence interval, now you are able to identify individuals who are at risk. So this will ultimately contribute to better diagnosis. And this would have not been the case if we had very wide 95% confidence interval. Okay, so after discussion about method, I am going to now talk about the findings and the insights that we have gained using subfield volumetry. I will start about age effect on hippocampal subfields. To start with, I'm going to talk about this meta-analysis, which that was the first step for me to characterize a trajectory of the subfields across the lifespan. The reason behind that is that majority of age-related studies on hippocampal subfield volumes are based on cross-sectional evidence and samples of limited age ranges that report inconsistent results. It was a challenge for us to gain insight to the true age trajectory of the subfields across the lifespan by qualitative review of the literature, particularly that evidence were different, studies were different in methods and sample characterizations. For the purpose of this talk, I'm going to just briefly discuss our finding on lifespan analysis. What you see here is a procedure of selection and screening for the papers. And at the end, we had 41 independent studies to be included in quantitative review, including 11 childhood study and 30 adulthood studies. So what you see here is the distribution of those 41 studies based on sample mean age that is indicated by field circles. And these horizontal lines are representing sample age range. Overall, this lifespan sample included more than 48,000 participants, representing ages of 4 to 94. So for me, the interesting question was that how reported effect sizes across these individuals varies as a function of sample mean age. We assessed that question where we included age mean of study sample as a predictor of estimated effect sizes for 41 studies. This dashed line is showing zero effect, and bubbles above is showing positive association between age and volume, and below is showing negative association. What we found was that independent of methodological differences, age mean of study sample negatively predicts estimated effect sizes, such that volumes were positively associated with age in samples of younger individuals, but that shifted toward negative association as age mean of a study sample increased toward later in life. This is consistent with nonlinear age trajectory, suggesting volume increase in development and then decline later in adulthood. I want to note that the observed age trajectory didn't differ across subfields. So this was first step, as I mentioned, but after all, it was based on cross-sectional evidence. So for us, the question was what is the trajectory of subfields in longitudinal sample, which is an essential research design for investigating of developmental changes. We asked that question in a large longitudinal sample with 474 healthy children and adults of ages 5 to 73, and with the follow-up sample of 189 participants. The delay between two visits were around 2.5 years. All the participants were recruited from Metro Detroit area and as a part of 2 ongoing independent longitudinal studies with the age ranges are specified here. All the participants were healthy and screened for the history of neurological, psychiatric diseases, or other clinical conditions. I already discussed the parameters for image acquisition. For hippocampal subfield segmentation, given that we had large sample size, we used automatic segmentation of hippocampal subfields or ASHA software that used a customized lifespan atlas. After segmentation, we quality check every single slice for potential error and correct it for severe errors according to procedure specified here in this study. For statistical analysis, we used latent change score modeling. That is a technique in structure equation modeling. To put it a simple way, this model is identical to calculating difference scores between time point 2 and time point 1, with a huge benefit of estimating change scores free of measurement error. Therefore, you have reliable estimates of mean change between 2 visits and also pattern of individual differences. Models were evaluated by an assortment of goodness of fit indices. So what you see here is spaghetti plots that are for HCV adjusted volumes against age of the participants. These lines are representing longitudinal assessment, orange representing volume shrinkage, and purple showing volume increase. And these are for other subfields. So for us, the first question was what is the volume change between 2 visits in respect to lifespan sample? So the result was non-significant, suggesting that volume is not changing between 2 visits. But that was not surprising for us because, as you see, there were significant differences in the pattern of the change. We were thinking that these positive and negative changes were averaged in respect to lifespan time window. And in fact, that was supported by significant variance in change, suggesting that there was substantial individual differences in the pattern of the changes in lifespan sample. So for us, follow-up question was if age of the participant is contributing to the observed variability across individuals and in predicting change pattern. So what you see here are scatter plots showing the result for these paths that age is predicting volume changes in lifespan sample. And this dashed line is showing volume stability between 2 visits, and that's about showing positive change below negative change. Age significantly predicted the change scores for all the subfields across the lifespan, such that younger individuals tended to gain volume, but that deaccelerated over age, across age, and shifted to volume shrinkage as age advanced toward 73. This is again consistent with nonlinear age trajectory, positive change, and then followed by negative change later in adulthood. As you can see, this is consistent with what we found in meta-analysis. However, this was a linear fit that is describing the change pattern, general change pattern across the entire lifespan. But still differences in change pattern across different stages of life were possible. So we assess that question. We define 5 age groups to represent childhood, adolescent, young, middle, and old adulthood, and assess volumetric changes within these 5 age groups. So what you see here is a mean volume change that is estimated as latent changes score for 3 ROIs and across these 5 age groups. But we found that volume of CA1-2 increased in young adulthood. That was interesting and surprising. Other than that, we found significant volume loss across old adulthood for all the subfields. This suggests that the volume loss, the probably age has a global effect in hippocampal subfield, and the effect is not specific. As you see that they didn't also differ in the timing because this effect was observed for the older individuals of 56 and between 73. Another interesting finding was that we found a significant variance in change, specifically in old adults. This is suggesting that although there is a general tendency for volume loss for old adults, but there are still some factors that impact the volume change pattern in old adults. So what I have described so far was that age is predicting volume differences or changes across lifespan. But question for us was, how do they inform us about memory development? So using longitudinal sample that I described, we asked that question. We used a word pair recognition memory task where individuals studied 26 word pairs, and after a minute of distraction, they completed test phase. I used just associative memory task in this testing phase where participants completed 16 forced choice trials. Half of these pairs were intact or already studied, half were recombined across word pairs. So memory performance was defined by the rate at which individuals correctly or incorrectly identified that pair as already studied. The reason that we use this associative memory was based on significant variability in this memory ability across lifespan, established relationship between this task and then the gyrus and CA3 specifically. That also is in accord with postulated memory processes supported by these subfields. We first confirm that memory ability changes across lifespan. So as you see, age was significantly predicting memory ability, memory change across lifespan. Younger individuals had greater gain or improvement between 2 visits, but that magnitude decreased across age toward stability between 2 visits later in adulthood. Here, I'm going to just note that our research design was longitudinal assessment, therefore required task repetition. In such research designs, we expect naturally individuals to benefit from task repetition. But as you see, younger individuals had greater benefit from that task repetition and less in older adult. That is consistent with reduced neural capacity. So we assess that question by including volume measures predicting memory changes. across all the subfields, only then the CA3 volume change significantly and positively predicted memory changes across lifespan, such that volume gain predicted memory gain and volume loss predicted memory loss. Importantly, this positive end of trend was driven mainly by individuals of younger ages, and negative end was driven by individuals of older ages. Again, other subfields didn't show this same association. I'm going to just highlight here that these findings provide further validity for volumetric investigations, because now method not only is able to detect nuanced individual differences, but also those volumetric changes are significantly predicting memory ability that are also in accord with memory processes attributed to those subfields. And at the end, I'm going to just briefly show some of our preliminary analysis from an ongoing study that we ask about sources of individual differences, such as sex, age, and blood pressure in old adults of ages 18 to 73. So we have 317 participant baseline and 152 at follow-up. So here I'm going to show you first the age trajectory for female by red and male indicated by blue color. Here you see age trajectory for linear, and bottom row is representing quadratic age effect. What we see in this analysis is that there is a significant differences in change pattern for between male and female participants for CA12 when we look at quadratic age effect. As you can see, younger male participants tend to gain volume, but that shifts to volume loss at around ages 45 and then 65. But later again, we see signs of or tendency for volume gain, which was interesting. And this trend was significantly different from the pattern observed for female participants. So for us, the question was if at all blood pressure is one of the factors can contribute or explain. So here, what you see is age is significantly predicting systolic blood pressure. So you could see that systolic blood pressure increases across age, similarly for male and female, with no differences in the pattern of the change. But when we look at systolic blood pressure in relation to change pattern control for the effect of age, we see that this higher blood pressure explains or contributes to volume gain in CA12, specifically in male participants. And that pattern is significantly different than what we observe for female participants. That was a very interesting and surprising result, because high blood pressure is considered as a risk factor, and therefore we expected volume loss. But here, we observe different pattern. There are some evidence in the literature that they report low blood pressure as risk factor in the individuals of very old ages. I mean, older than 75. But our sample didn't include those old ages, and was again interesting that we observed a similar pattern in this sample. Nonetheless, we think that to be able to meaningfully interpret this result, we need to also look at the memory indices and other indices to better describe cardiovascular risk factors. And this is I want to conclude the talk by talking about a little bit about the method. In this talk, I provided the evidence for validity, reliability, and sensitivity of the volumetric assessment that all characterizes a great measurement and method. However, there are some inferential considerations when interpreting the result. As we know, volumetric measurements are indices of structural underlying neural mechanisms. And all we can do is speculate, and it is hard to really know what is going on. Specifically, that might be challenging if you have very large sample representing different ages, because volume measures can't represent different neural processes. For example, volume decline in younger ages may show or indicate pruning, neural pruning. That is a positive change, but later in life may represent neuronal loss or demyelination or neuropil shrinkage or then neuronal shrinkage. And same also goes for healthy and clinical population. Volumetric measures in healthy populations may indicate different neural processes. For example, volume loss in healthy population may show, again, neuronal shrinkage, but in clinical population, that might be an indication of neuronal loss. However, I want to just highlight that there are some approaches that we could use to increase our understanding and enhance our understanding. One of them is to include memory indices and pair it with our volumetric investigations. We could also gain better insight about the structure of tissue by pairing volumetric measures with other neuro imaging modalities, for example, quantitative MRI. Using blood-based biomarkers is another approach to, again, understand the sensitivity or vulnerability of subfields. And another approach is that we could pair MRI technique with postmortem studies. For example, we could have MRI measures in living population and then continue to do postmortem studies on the same individuals. This approach is specifically enables us to better understand the measures and investigation on both sides. For example, volume measure, we could better interpret postmortem studies, and postmortem studies enable us to better understand volumetric changes. And at the end, I am going to thank the wonderful people that I am working and I have been working. One and first is my postdoc mentor, Dr. Anna Doherty, who was my also PhD co-mentor. And also I want to thank my previous PhD mentor, Dr. Noah Fenn. I want to thank all the funding resources, the current and previous lab members, our participants that make this happen, also Neuroimaging Corps for giving me this opportunity, and thank you all for listening.
Right. Thank you very much, Roya. I think we do have some questions coming in right now. You can also see in the chat that I just put the event evaluation, so I would really appreciate it if you could all take the time to fill out the evaluation before you sign off. I will also be sending it out tomorrow in a follow-up email as well. One question that we have, it says, the impacts of blood pressure on the volume of the hippocampus is very interesting. Would different vascular patterns explain these differences in CA1, CA2, and other zones?
So what I know that, again, the pattern of vascularization in CA1 is a little bit different. It is supplied by large brain arteries. I think ventral intrahippocampal arteries. And that actually makes CA1 to be prone to more prone to vascularization. So because as I showed earlier in my presentation, CA1 is the region that is highly active and it actually has higher energy demand, but at the same time, it is less vascularized. So when you have like possibility of ischemia, that might be the case in high blood pressure, you expect to see actually opposite effect in CA1. So as you said, this is interesting result for us too. And the fact that we found opposite effect, or at least in volumetric measurement of these findings, we see beneficial effect of that. Again, we need memory indices to interpret that. But probably one explanation is that like blood pressure effect differs as a function of age. But again, we are in the process of exploring this data. And Anna is an expert in this field. I'm not sure if she wants to comment anything about this. Okay.
Anna, I gave you the mic if you want to say anything.
Thank you. I really appreciate it. Roya explained it really well, that we do think that there are particularly vulnerabilities in the CA1 region. However, we could also posit moving into also CA3 dentate gyrus, and that perhaps the type of vascular risk and the timing in the lifespan of their vulnerability might help differentiate some of these mixed decline states in typical aging, and then even more so maybe help us differentiate early detection for vascular dementia as compared to other sources of dementia. So stay tuned. The Detroit Aging Brain Study is trying to answer that question right now.
Awesome. Right. Any other questions for Roya? Yeah. Yes, Valentine, you have a question.
Yep. I'm wondering whether we found differences in racial differences as regards hypertension on hippocampal volume in the participants. For example, African Americans and then others as it relates to hippocampal volume in hypertension.
Mm-hmm. That's a great question, and it is in our agenda to look at the racial differences, but we haven't looked at it yet, so I cannot answer that question.
All right, I have another question in the Q&A. It says, thank you for your talk. You presented your manual segmentation procedure, and I was wondering whether you compared your procedure to automated segmentation algorithms of hippocampal subfields.
So like our ashes, you mean, software? If it is the case, there are some like paper that actually they compare. Not us, but yes, there are some comparisons, some evidence that they look at.
This is yes or free surfer?
No, not free surfer, but for the protocol that you're using for ashes, yes. And but free surfer actually is a method that has a very big concern in volume estimation. For example, it is using, I believe, the algorithms based on the body of the hippocampus to estimate the volumes on the head and tail, which is very complicated. We have a lot of folding and also twist on the two end of longitudinal axis of the hippocampus. So there are actually evidence showing that there is some questions about the validity of those estimates. Specifically, I know that CA1 is although is larger anatomically from CA2 and 3, but in free surfer is actually is estimated smaller. So it is, again, it has raised this concern that if we could, or we should use a free surfer in our segmentations.
Do you mind if I jump in a little bit too?
Yeah, please.
Oh, great. So I think part of this question might be coming from our thinking about how do we measure whole hippocampus volume. And in general, there's a great amount of consensus in the field at large of our definition of the hippocampus structure and how we might measure it from MRI. And so we often are interested in automated versus manual and trying to think about how can we automate these methods. It might not be obvious, Roya, to others that the hippocampal subfields, there's currently no consensus on the definitions of these subfields. And so every protocol that does it a little bit differently. Many of us have more similarities than not, but it's not as simple as picking up someone else's automated method and then comparing it to another manual method, because by definition, a lot of those boundaries are just different. And so it's not really a difference of automation versus manual. It's just fundamentally a different method. And so, Roy, I wonder if you might talk about some movements in the field of trying to address that so that we could create more standardization.
Yes, I had actually this slide for
Oh,
that.
great. There you go.
Yes, I mean, as you said, the abundance of the protocols in the field has made it very challenging to compare across the findings. As Anna said, we have different protocols, some of them, in the number of the ROIs defined, which ROIs you want to combine or group together, or which part of the extent that you want to estimate the subfields on the longitudinal axis. But for example, our protocol is in body of the hippocampus, some are on the full longitudinal axis of the hippocampus. So to address that concern and to define a harmonized protocol, this group, the hippocampal subfield group, is actually now trying to introduce a protocol that I have put an example from segmentation on the body of hippocampus, that they are doing a lot of efforts to validate, and also they are assessing the reliability of the assessment. Ultimately, we not only have the method that is validated and also reliable, but we also can now use in the field across labs. So therefore, we will be able to compare across the findings. As you see, this method, for example, now has separate segmentations for CA3 and CA2. In the context of, again, clinical investigations, it is also offering the opportunities for the investigators, if they want to have sensitive and specific, biomarker of any clinical conditions.
Hey, any other questions for Roya before we conclude today? All right, well, with that, I think we'll just end a few minutes early. Please join us next month. Our next neuroimaging summer series will be on June 7th. It's a Friday, and we'll have John Frederick Nielsen, Naveed Siraji, and Amaya Murgua. They will be presenting on myelin content with MRI, existing and emerging methods. So should be another interesting talk, and we hope you can all make it. All right. Take care, everyone.